there, and welcome to this month's StemBox tutorial, where we're going to be talking about everything sharks. Every experiment in our box today is going to be an experiment that researchers aboard an ocean vessel actually do to learn more about sharks, their habitat, and their biology. So stay tuned because we're going to dive right into our first experiment. So on most research vessels, the first step is getting the shark. And what actually happens is that most boats will have a sort of lift. So they basically scoop the shark up out of the water, bring it on board, but the team only has 15 minutes to work on the shark for reasons for safety of the shark and safety to the crew. Then the next thing they do is they go ahead and they start taking measurements. This would be something one crew member does, while the other members are drawing blood or running the other experiments and going to run through today with us. So the first thing we need to do today is we have our shark Ruby. You need your pencil, you need your shark map, and you need your tape measure. So we're gonna go ahead and say that we've caught Ruby and she's blindfolded, so she doesn't feel very scared or anything. And we're gonna be very quick about this for her sake. So we're gonna get our stuff ready, and then according to our map, there are several pieces of our fish that we need to measure. We have the dorsal fin length, nose to tail circumference, second dorsal fin, pelvic fin, so many fins, pectoral fin, and a caudal fin, which is basically just a tail fin. And you can find all of these parts on your protocol, and then you can correlate them to your shark. So basically, I'm just gonna go through and measure Ruby as it would specify on our protocol, but in centimeters. And that's the side of your tool that has more ticks on it. So basically nose to tail and just to the tip, it's about 27.5, and then I'm going to do what researchers do, and they write that in. And we're gonna go through each part of Ruby and do that as quickly as possible, and we're gonna practice getting faster on it. So time yourself and see how much faster you can get about doing this. And then as you go along, make sure that you're recording everything. The reason the researchers need to measure their sharks is so that they can understand how the sharks are growing in their environments. So if they have a bunch of great white sharks and one of those sharks starts to look a little small, and then there's a population of them that are all a little bit smaller than what is average for a great white shark, then they have reason for concern, and they need to start looking into what's going on in that shark's habitat or its life cycle that might be damaging its population. This is why it's so important for our researchers to know about our individual sharks' body measurements. So once you've finished measuring your shark Ruby, go to the StemBox website and compare your answers to ours. We want to see what kind of standard deviation we see between all of our StemBox's answers, so make sure to include your measurements if you send us an email, or on your Instagram, or Twitter, or Facebook. The second experiment in our box replicates the tagging that researchers do on boats to learn more about the shark's movement, the shark's habitat, and how quickly a shark might recover after its research. So what we're gonna do is we're gonna go through one by one what each of the tags means, and we're going to place them on Ruby to understand better what she does. If you're interested in learning about more of the tags that researchers use besides these 4 main ones, go to our website and you can find all the links to the different research universities or groups out there that do this kind of research. For this experiment, all you need is your shark Ruby, you need your 4 tags, and you need your protocol so that you understand where to place all of these tags. The first tag we're going to apply to Ruby is the spot tag. The spot tag is the only tag that allows researchers to see in real time where Ruby is going. So the first thing we have to do is make sure that she is comfortable, of course, and then acting as our tag, our spot tag, we're going to clip it to the top of Ruby's dorsal fin. The reason this needs to be on the dorsal fin is that the antenna for this tag has to be as close to the surface of the water as possible, because the only way that signal can transmit is if the antenna is above water for longer than 30 seconds. So it needs to have that break of the surface to be able to send a signal out to a satellite, and for that satellite to send back its coordinates to any researcher who is tracking the shark. But the information about sharks' whereabouts in real time is actually some very powerful data that researchers need to keep under wraps. Sharks, some species, are considered vulnerable, which means that they're being hunted and they're being caught in nets, and it's very important that fishermen who are out to get these sharks don't have access to the whereabouts of these sharks in real time. That would be a disaster. So researchers keep this stuff tightly under wraps, and any data that we have is old, so we don't really necessarily know that our sharks are just in that location. Our second tag is called the Accelerometer Tag. Now, when you think of accelerometer, it sounds basically like the exact same thing that is in your phone. And in your tag, it is the same mechanism. So when your phone, you're Snapchatting and you take a picture while you're driving, you're not driving, your mom's driving or something, and then you can switch to the one that says miles per hour, that's the accelerometer on your phone working. And in the same way, this tag can track the miles per hour that the shark is moving. It's a really great way for researchers to understand how well a shark is recovering after it's been tagged, if it's swimming away at a good rate, and what kind of migratory pattern it is. So maybe in the winter, a shark might swim a little bit slower and they can tell with the accelerometer. In the summer, maybe it's having a fun time and it's hunting a lot more. The accelerometer can tell you how fast that shark is going. So this tag goes directly underneath the spot tag, right there on the dorsal fin as well. Our third tag is the PSAT tag. This tag will be clipped onto the dorsal fin below our spot tag and below our accelerometer. So the way the PSAT works is that it will pop off. The signal picked up by this PSAT will be picked up by a satellite, can tell the researchers a lot about a shark's habitat and where it's been hanging out. So things like the depth of the ocean, the temperature of the ocean, and it can also tell you a lot about the seasonal mating habits and migratory patterns of this shark, but it's kind of a one-shot deal, so you only get one pop-off. Our fourth tag is the Acoustic Tag. Now this tag is special. It gets implanted in Ruby's belly, just right under the skin. So for us, we're going to just do a tiny little clip on her belly. And don't worry, this doesn't hurt the sharks. It's very much like you getting a shot or your dog getting microchipped. The way the Acoustic Tag works is it's very dependent on a receiver. So what this does is it sends out a sound underwater, like a bing, 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 whenever the shark is swimming by. And just like sonar, it needs to bounce off of something, and that would be the acoustic receiver that's planted by the researchers. So there's tons of these all over the ocean, depending on where the researchers want to know about. And what happens is when the shark swims within a kilometer, usually it's a kilometer, the ping is detectable by the receiver. And the data that comes back from that can tell researchers about where the shark has been swimming, what it's doing, where it's, like how deep it is, all sorts of stuff that might be really helpful for researchers to understand the habitat, biology, and movement of sharks. Our third experiment today is focusing on cartography. Cartography is the study of maps, specifically latitude and longitude, which can come together to form coordinates like a grid. So this data is a direct result of the spot tag on the dorsal fin we put on Ruby. In our case though, we're going to be using real coordinates from a shark named Mary Lee that the OSearch team has tagged in the past. So if you want to check out your answers and make sure that you've gotten results that match up with the real data on the OSearch website, go to www.osearch.org and you can track all the other sharks they've tagged. There are so many, all kinds of sharks too, and you can see pictures and meet them, and it's super fun to go on and to use the rest of your dots to plot on your map. So what we're gonna do is we're gonna get started, and we have a couple of coordinates here. We're gonna get you started with the first few. We don't need Ruby right now, she can take a nap, but we're gonna take our coordinates, our pencil, and our dots to our map, which you will have in your box folded up. So the first two things you need to know about cartography are there are two components to cartography. There's latitude and longitude. So the best way to remember this is that longitude, think of somebody who's long, tall. They stand straight up and down, basically like the up and down lines on the map. That's longitude. Latitude sounds a lot like somebody's laying flat. They are latitude. Latitude is this way on the map. It's the horizontal line. So we're gonna go ahead and plot our first coordinate. The first thing we're gonna look at is the latitude, which is 34 north. That means we go to 30 and 40. And somewhere between that, about 4 ticks up, we go to 34. Then we need to see our longitude. Remember, that's up and down. And that was west 77. So that's here, and that's in between 80 and 70, mostly on the 80 side. So I'm gonna follow this finger line down till I get to where my other finger is here. And that's right off the East Coast. So to mark my spot, I'm gonna go ahead and put our first dot. So that's our first spot tag signal for the shark. We're gonna go ahead and skip to the 4th dot because we want you guys to see the difference, but we don't want to give it all away for you. So our 4th coordinate is 29 north and 81 west. And this is gonna look really close together because this map is a map of the world, and this is just one shark off the coast of America. So you can use this map to track other O-Search sharks when you're done with this one. So that was, again, 29 north, 81 west. So a little bit below 30 and above 20 a lot. And then 81. So that is basically right by Florida. So you can see that Mary Lee's migratory pattern is already starting to take a little bit of a shape. 2 data points are not necessarily a data set. But you guys can see how this works. Follow the rest of your coordinates and check your answers on the O-Search website. You can compare all these movements to what they have listed for the shark named Mary Lee on their drop-down picker of sharks that they've worked with. If you want to learn more about this, go to our website. You can go to O-Search's website. You won't be able to get exact coordinates because, for reasons we talked about earlier, a lot of this data is very secret to protect these sharks. There's only generalized patterns that you can look at, but you can still replicate them and add them to your map if you have a favorite shark as they occur. Because this team is always collecting data and always learning about those sharks. It's crazy. Our final experiment today explores the biology of a shark's eye. So there's something called tapetum lucidium that makes sharks such an amazing predator in dark water. This is something that makes the back of a shark's eye reflective. The need for a shark to have a reflective surface on the back of its eye is so important. The way it works is the light comes into the eye, it hits the back of the eye for the shark, the shiny part, and like a mirror, that light is reflected back out of the shark's eye, so now it's even brighter in front of it. But for humans, what we have happen is the light goes directly back and stops. It projects the image and that's all we have. But this is kind of a built-in flashlight for sharks. In fact, your cat or your dog has tapetum lucidium as well. Next time you're at home and it's dark or there's a flashlight around or you're outside and you see a cat, if you see its eyes glowing like orbs, that's tapetum lucidium. It's basically just a shiny coating on the back of the eye. Humans don't have this. And the experiment we're going to do today is going to demonstrate the difference in reflective properties of these types of eyes. The pieces you need for this experiment today that come in your box are glue, your LED flashlight or just the regular flashlight, construction paper that's blue, construction paper that's black, and some foil. You will also need a pair of scissors to cut these to size, so have a parent around and help you if they're sharp. Always hold scissors properly, don't run with them. Okay, let's get started. The first step in this experiment is we have to get our background ready. So basically you're gonna have a piece of black construction paper in your box. It may or may not be folded already, but go ahead if it's not and fold it hamburger style so it's flat like this and make a nice solid crease. Okay, now that that's done, the second step is we're going to cut out a piece of blue square that'll fit on the top half of this construction paper. So something about that size, it doesn't need to be perfect. And then you want to get something cut out of the foil that is comparable in size so that we don't have too many variables in this experiment to your blue square. And we have 2 squares that look very similar in size, blue and foil, right? So we're gonna open this up and kind of like a greeting card almost, we're gonna go ahead and use our glue stick to glue these onto the paper. And then our second one, so your final product should basically open up like a card, and you have a human eye and a shark eye. Now the next part is going to tell us which one is better at seeing in the dark. The important thing to notice about the construction paper is when you shine the light on it and you look at its reflection on the black paper or even just out into the world in general, there's really not a lot going on there. The construction paper is absorbing most of the light, just like a human eye would. But when you shine it on the shark eye or the foil, there's a whole other flashlight coming at you basically, and you can really notice the difference on the black paper. So again, so down here, shark eye, human eye, shark eye, human eye. There's really not a lot of reflection happening at all in the human eye, but the shark eye is like a second flashlight coming right back at you. So now that you know the difference between a shark eye and a human eye, it may seem like sharks and humans have nothing in common. But in fact, we do have a lot in common. Both of our survival is based on the conservation of ocean wildlife and the ecosystem within the ocean. Sharks are a huge part of that ecosystem, and so it's really important that this kind of research about where sharks are living, how they're migrating, what their habitats are like, if they're healthy. This research needs to be done so that we learn how to keep our oceans clean and safe, and a main part of that ecosystem, the shark, safe as well. But there are things at home that you can do to make sure that the ocean is clean, that sharks are happy, and our researcher in the next video is so excited to tell you about that. Danielle Carter from the Seattle Aquarium is an amazing resource. She can tell you all about sharks and all about the things you can do at home to keep the ocean a super happy, healthy place. So go to our next video to learn more about that and everything sharks. Thank you all so much for following along with this month's box. I can't wait to hear what you all think about this. Please send us your photos, any videos you have of doing this experiment. I really hope that you learned something about sharks today that you didn't know before, or you maybe want to be a marine biologist now. But even if you don't, that's okay. Thank you again so much for watching. This has been Kina and Ruby the Shark. Catch you later.
Caudal fin, that is where they get their power from. So the faster they move that, the faster they're gonna go in the water.
Okay.
And if you look at how a shark's body is shaped,